Hi everyone, in this presentation we are going to see about the classification of enzymes and the mechanism of enzyme action. The first we are going to see about the enzyme biological catalyst, usually proteins that speed up chemical reaction in living organism. They work by lowering the activation energy required for a reaction to occur, allowing the reaction to proceed more efficiently and rapidly. Enzymes are highly specific, meaning each enzyme typically catalyzes only one type of reaction or acts on the specific substrate. Enzymes are essential for various physiological processes such as digestion, metabolism, and DNA replication, among many others. They operate by binding to their substrate at an active site, forming an enzyme substrate complex which helps the reaction occur. The next one is classification of enzymes. Enzymes are classified into six main categories based on the type of reaction they catalyze. First one is oxidoreductases. These enzymes catalyze oxidation reduction reaction when electrons are transferred between the molecules. For example, dehydrogenases, oxidases, and reductases. The second one is transferase. These enzymes catalyze the transfer of functional groups between molecules. For example, aminotransferase, methyltransferases, and kinase enzymes. The third one is hydrolysis. These enzymes catalyze the hydrolysis of bonds where a water molecule is used to break a bond. Example, lipase, protease, and amylases. The fourth one is lysis. These enzymes catalyze the breakdown of chemical bonds without the addition of water, often resulting in the formation of a double bond or ring structure. For example, decarboxylase, aldolases, and dehydrogenases. The next one is isomerases. These enzymes catalyze the conversion of molecules within the same molecule, rearranging atoms or groups such as converting an isomer to another. Example, epimerases, racemases, and isomerases. The sixth one is ligases. These enzymes catalyze the joining of two molecules with the formation of a new bond, typically with the use of energy derived from ATP. For example, DNA ligase and synthetases. The enzyme are substrate complex. It moves to the formation of enzyme or product complex. Finally, the product leaving the active site of enzymes. The first one is in this process the substrate binding. The enzymes have a specific region called the active site where the substrate binds. The active site shape is complementary to the substrate, ensuring specificity. The second one is formation of enzyme substrate complex where the substrate binds to the enzyme's active site and enzyme substrate complex is formed. This complex stabilizes the transition state and lowers the activation energy needs for the reaction. The third one is the catalysis. The enzymes facilitates the conversion of the substrate into the product by weakening existing bonds, stabilizing intermediate states, or binding reactants closer together, depending on the reaction type. The fourth one is product realized. Once the reaction is complete, the product has a different shape or lower affinity for the active site, so it is realized for the enzymes. The fifth one is enzyme recycling. The enzyme is unchanged after the reaction and can bind to a new substrate to repeat the process. The next is types of enzyme mechanism. The first one is lock and key model. The substrate fits perfectly into the enzyme active site, much like a key fits into a lock. The second one is induced fit model. The active site undergoes a conformational change upon substrate binding, enhancing the enzyme's ability to catalyze the reaction. The third one is covalent catalysis. Temporary covalent bonds are formed between the enzymes and substrate during the reaction. The fourth one is acid-base catalysis. The enzyme donates or accepts protons to stabilize the transition state. The fifth one is strain or distortion. The enzymes applies strain on certain bonds within the substrate making them more susceptible to breaking. This picture shows about the types of enzyme mechanism. Thank you all.